Let's begin with the seminar today. Cell division, a phenomenal thing that happens in nature, is of three types: amitosis, mitosis, and meiosis. Amitosis is a simple kind of division where the cell simply divides into two. In the last class, our classmates explained us about the mitotic cell division. Today, me and my team, we are here to explain you all about this beautiful thing that happens in nature, meiosis. Okay, can I take a simple analogy here? So when we go to a movie, the movie is divided into many parts, like first half, interval, the second half, and the climax, right? Like that, in meiosis also, meiosis 1, the interval called as kinesis, meiosis 2, and the cytokinesis process. Is this analogy easy for you? So let's begin. So before beginning into meiosis, I'm going to tell you some subtle differences between mitosis and meiosis. Mitosis is a somatic kind of division which happens in somatic cells, whereas meiosis happens in germ cells. These germ cells are cells that are precursors to the gametes in case of humans. And also a very important point that we all should note here is in mitosis, the number of cells they increase, but the number of chromosomes they do not increase. It is equational division. In the case of meiosis, it is different. The number of cells they of course increase because it is division, right? But the number of chromosomes they're going to get reduced. The diploid parent cell will produce haploid number of daughter cells. This is very, very essential. In the end of the presentation, you'll also know why is this essential. Before beginning our presentation, I want you all to look into this beautiful video.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is production of 4 haploid cells, each with a single chromatid from each chromosome. When haploid sperm and egg unite during fertilization, the resulting cell is diploid, having received 1 chromosome of each pair from each parent.
Interesting, isn't it?
Meiosis is a
Actually, this
special
is a real footage of microscopic footage of how meiosis happens. Meiosis was first discovered by a German biologist named Oscar Hertwig in 1876 and was first described in sea urchin eggs. So, without any further ado, here comes Ashrita to explain you all about the prophase 1 stage.
The prophase 1 is the first and the longest stage of the meiosis. It is divided into 5 substages, which is lipotene, then zygote, the third pachytene, the fourth is the diplotene, and the fifth, the last stage is of diakinesis. Liptoten in Greek means fine threads. During this liptoten, the chromosomes they begin to condense and they become fine visible fine threads. So they have marked increase in the nuclear volume. The zygote is a chromosomal pairing of occurs forming bivalents. So homologous pairing of chromosomes is also called as synapsis. Packaging is this step which is very important for the process of crossing over and 2 homologous chromosomes. These begin to exchange their genetic material forming a tetrad. Diplotin, the chromosome uncoils the transcription to occur and slightly move further from each other. The diakinesis is the last stage of prophase 1. So during this stage, the chiasmata terminalizes and the homologous chromosome separates. Nuclear membrane and the nuclear disappears at the end of the prophase 1. So next comes Ramya to say about metaphase.
So guys, here I have to explain about metaphase one. In this metaphase one, the chromosomes will appear in the sorry. So guys, here we have the second stage of meiosis one called metaphase one, where we can clearly observe the chromosomes in the microscope. This is a stage where all the homolog chromosomes get arranged in the center of the cell. This is called metaphase plate. Also, the spindle fibers get themselves aligned in the equatorial plane. This will help them to attach the spindle fibers in the centromere of the chromosome. Typically, the chromosomes line up in a single row on a structure called metaphase plate. However, in humans, the chromosomes in the metaphase may not always align in a single row on the metaphase plate. This is all about metaphase 1. Now comes the interesting stage called anaphase 1, where the spindle fibers begins to pull the homologous chromosome apart from the metaphase plate to the poles of the cell. A very important point to note here is that sister chromatids remains attached to each other in this anaphase 1 stage. Thank you. Now comes here to explain about telophase.
Hi everyone, I am here to explain two phases of cell cycle, telophase 1 and interkinesis. In telophase 1, the chromosome which are disappear will come again, especially nucleolus and nuclear membrane. This nuclear membrane will bound the chromosomes and cytokinesis appears, which divides the cells into two diagonal cells. These cells are called daughter cells. These cells are 23 pairs of chromosomes. This is because in anaphase 1, the sister chromatids will remain attached to centromere. On following this, it is followed by interkinesis. It is also known as interphase 2. In this phase, uh, in this phase, no DNA replication will occur. This is known as it is because S phase will not uh, S phase will not happen this interkinesis. Here comes Kavya to explain meiosis 2.
I think now half of the movie is over and interval is also over. Now comes the second part and not very interesting. Like it's simple mitotic division. It's called as meiosis 2, but it's simple mitotic division. So in meiosis 2 we have prophase 2. Here the nuclear envelope they break down. The nucleolus they also disappear. So a very important point I want you all to note here is in prophase 1 we had a very important stage called as crossing over. It happened in Pachytin stage. Do you all guys remember? In prophase 1 we had Pachytin stage, then crossing over occurred, right? DNA recombination occurred. But in prophase 2 it's not going to happen. It's not going to happen. DNA recombination will not happen. And this is very important because two times should not recombine, right? Already the prophase 1, the DNA got recombined. The prophase 2, it's just going to divide. And in prophase 2, there are two cells. Can you see the slide? There are two cells. And the first cell and second cell, we are going to have, in case of humans, 23 pairs of chromosomes. 23 pairs. So first 23 pair, second 23 pair. And after that, the spindle apparatus, they'll also get formed on the poles. Next is the metaphase 2, where all the chromosomes, they'll get aligned in the metaphasic plate, the center, and the spindle fibers, they'll get arranged in the equatorial plane for them to catch the chromosomes in the centromere. And also, the next stage is anaphase 2, where the spindle fiber they'll pull out their threads from them and they'll catch the chromosomes in the centromere, and from that they'll just pull the chromosomes apart. From now, what will happen? The 23 pair, they'll get divided 23, 23. So first it was 23 pair. Now it's going to be 23, 23. Can you understand what I'm going to coming to say? So in each cell now it will be 23, 23, 23, 23. The cells will be haploid in number. Next, now the chromosomes are individual. And next is the last, like telophase 2 phase, where again the nuclear membrane they'll reappear. In prophase 2, it was disappearing. Now it will enclose the chromosome and it will form the nucleus. The nuclear membrane, the nucleolus, all the other organelles, all of them will reappear. the chromosome will become distinct, like indistinct, sorry, indistinct, and we can't like see them in the microscope. In the metaphase, we can like easily observe them like chromosome shape, egg shape, but now we can't see them. It will become indistinct in telophase two, and now comes the climax. Like everybody they are in the two cells. Now the climax is they're gonna divide. This is the breakup story. The cytokinesis. In the cytokinesis, the cell will divide and now it will have four haploid cells. And I think the movie is over and here comes Bharat to enlighten us with some key takeaways so that we can remember some important points in meiosis. And also, he has some interesting quiz to ask you all. So be ready.
Actually, yeah, this is Bharat here. So, the first point is like meiosis is also said to be reduction division, right? Yeah. Then, because the daughter cell is nearly half of the number of chromosome from their parent cell, so it's said to be like meiosis is a reductional division, and um, the diploid number of species is nearly constant, and the crossing over paves for new gene combination. Yeah, I'm really sorry. Yeah, actually, we can start the quiz right here. So it will be a little interesting. Yeah. So when does crossing over occurs? Yeah, could you please answer? Anyone? Yeah. Yeah. Packaging, that's correct. But yeah, that's correct. In Profess one, especially in packaging, that's correct. And how many times DNA replication occurs in meiosis? Okay. One. That's right. And what are the egg-shaped chromosome that forms? Sorry. Okay. Gasmata. And then why the daughter cells are not identical to the parent cell due to crossing over. Yeah. And then when during the meiotic division do not sorry do the centromere divide. Anyone answer? Could you please stand up? Anyone stand up and answer? A single answer. Yeah, I need. Anaphase? No. Anaphase 2 is the right answer here. Homologous pairing of chromosome is called homologous pairing of chromosome is called synapsis. Yeah, well, thank you so much.